Introduction. Statins are drugs that help to lower cholesterol level in the blood and help to prevent coronary heart disease in patients without a history of cardiovascular diseases or those who are at a very high risk of developing CVD or have had some form of CVD. Pharmacological action. Statin inhibits HMG-CoA reductase in cholesterol synthesis, therefore cholesterol synthesis decreases and increases the expression of LDL receptors. Higher amount of receptors cause increased LDL uptake by the hepatocytes and cause decreased LDL cholesterol in the blood. Statin also reduces blood level of triglycerides and slightly increases level of HDL. Types of statin. Type 1 statin have butyric groups such as lovastatin, pravastatin, and simvastatin, while type 2 statins are fully synthetic and have fluorophenyl group such as fluvastatin and atorvastatin. For example, the difference in structure in different types of statins.
In 1971, a Japanese biochemist, Akira Endo, worked with a pharmaceutical company named Sankyo. Endo and his team began the search for a cholesterol-lowering drug which is an inhibitor for HMG-CoA reductase. Later, they identified a molecule named mevastatin produced by a fungus named Penicillium citrinum to be the inhibitor of the enzyme. In 1980s, an American medicinal chemist, Bruce D. Roth, synthesized a compound called atorvastatin, which is more potent and displayed fewer side effects compared to other statin drugs.
Drug discovery. In New York, Endo was surprised by the large number of elderly and overweight American people compared to Japanese, and this experience of living in New York made him realize the importance of developing a cholesterol-lowering drug. In 1960, a number of lipid-lowering agents were available, but none of them were safe and effective. After that, this is what had happened. Meanwhile, Merck Pharmaceuticals isolated a related CMPD lovastatin from the fungus Aspergillus, and this is what happened next. Meanwhile, Sineo and Scripps were entering clinical trial on another statin, which was pravastatin. Soon after, Merck came out with a second statin, which was simvastatin. As for simvastatin, it is produced by chemical modification and approximately twice as potent as pravastatin and lovastatin, and monasterate is another source of lovastatin which is red yeast rice. Soon, several synthetic joined the groups of natural statin called type 2 statins, which were as follows.
In drug design section, we will focus only on atorvastatin. This is atorvastatin, developed by Bruce Roth in 1997. Why atorvastatin? Because it is the most potent statin after rosuvastatin, and it is the highest selling drug of all time. Lead compound development of atorvastatin, pyridase system is the starting template and then undergoes lactonization in base hydrolysis, but the resulting compound possesses only 1/10 of the potency of mevastatin. To overcome it, bromine and chlorine were added to the most potent analog, but the resulting compound displays considerable toxicity, so the cycle addition of S-lactones and acetylenes were conducted. At last, the best activity is found in the 3-phenyl 4-carbosamidophenyl analog because it possesses almost the same potency as simvastatin and lovastatin. Finally, atorvastatin was developed as pure stereoisomer to avoid the disadvantage in potency and metabolism. Several chemical reactions were improved later, and preclinical studies were conducted to file an investigational new drug application with FDA. For preclinical dosing trial of atorvastatin, it was found that atorvastatin is more potent than marketed statins at that time. In addition, it was also found that atorvastatin is highly protein-bound and has a long plasma half-life. After several trials, it was found that the maximum recommended dosage of atorvastatin is 80 mg per day for a single dose.
In vivo and in vitro preclinical trials, safety and efficacy testing. For in vitro preclinical testing on statins, statins are administered orally. Therefore, one of the in vitro testing is to study the effects of statins on microorganisms infecting oral and perioral tissues. From the studies, it has been proven that statins have antimicrobial effects on oral microorganisms. For example, simvastatin is most effective against the periodontal pathogens and dental plaque bacteria. Other statins also exhibit antiviral and antifungal properties. In vivo trials on animal models are used to test for statins' efficacy and toxicity. There are several preclinical testing on statins involving animal models. First, dosage toxicity studies on rats, mice, mini pigs, dogs, and monkeys. Second, reproductive toxicity studies on rats and rabbits. Third and fourth are on rats and mice, which are mutagenicity assays and carcinogenicity studies. For the choice of animal models, statin lowers the plasma triglyceride rather than cholesterol in rats. However, triglyceride lowering is correlated with cholesterol lowering. Therefore, normal rats can be used to assess the efficacy of statins. A study shows that atorvastatin is more efficacious than lovastatin in rats. Some statins show adverse effects on animal models such as degeneration of muscle fiber, minor morphologic changes in testicular tissue, GIT erosion and hemorrhages, bleeding in the brainstem and lens opacity. However, the most toxic statin, cerevastatin, had been withdrawn from the market. Other statins have been proven to cause no primary embryonic or teratogenic effects, and there is no evidence that statin had any mutagenic effects. Therefore, from these preclinical trials, it can be concluded that statin had been proven its safety and efficacy.
Atorvastatin has gone through 4 clinical trials before getting the approval from the Food and Drug Administration (FDA), namely ASCOT, CUTS, TNT, and IDEAL. The objective of ASCOT and CUTS trials are basically the same, that is to evaluate the effect of Lipitor on cardiovascular disease endpoints. Also, they share the same study requirements, such as both trials were double-blinded. 10 mg of Lipitor was used against placebo, and the condition is that the participants do not suffer from cardiovascular disease. The only difference is that ASCOT trial is conducted on hypertensive patients, while CUTS trial was one for diabetes. According to the ASCOT trial result, Lipitor reduced rate of coronary events with an outstanding 36% risk reduction. Besides, it also reduced risk of vascularization by 42%. As in for the CUTS trial, Lipitor was proved to decrease rate of major cardiovascular events with a relative risk of reduction of 37%. It also significantly reduced the risk of stroke and myocardial infarction by 48% and 42% respectively. To sum up, both ASCOT and CUTS trial were a huge success in proving that Lipitor is effective in treating cardiovascular events.
TNT stands for Treating to New Targets. The objective is to compare the effect of Lipitor 10 mg and 80 mg daily in patients with stable coronary heart disease, involving 10,001 patients, men and women 35 to 75 years of age who had clinically evidence of coronary heart disease. Results. The mean LDL cholesterol levels were 2 mmol per liter during treatment with 80 mg Lipitor and 2.6 mmol per liter during treatment with 10 mg. Reduction in the rate of major cardiovascular events of 2.2% and 22% relative reduction in risk. There was no effect on mortality. Conclusions. The investigators concluded that intensive lipid-lowering therapy with 80 mg of Lipitor per day in patients with stable coronary heart disease provides significant clinical benefits beyond that afforded by treatment with 10 mg per day. IDEAL stands for the incremental decrease in endpoint through aggressive lipid lowering. Objective to investigate whether high-dose Lipitor daily would yield incremental benefits compared with simvastatin in patients with previous myocardial infarction. Involved 88,888 patients, men and women aged up to 80 years old with a history of myocardial infarction. The mean LDL cholesterol levels were 2.1 millimole per liter during treatment with 80 mg Lipitor and 2.7 millimole per liter during treatment with 20 mg simvastatin. Lipitor modestly reduced major cardiovascular event by 1.7%, any coronary heart disease event by 3.6%, in non-fatal myocardial infarction by 1.2%. There was no reduction in coronary heart disease death or all-cause mortality. Lipitor at 80 mg daily was less well tolerated than simvastatin, but there was no myopathy reported. Conclusion. The investigators concluded that patients with myocardial infarction may benefit from intensive lowering of LDL cholesterol levels without an increase in non-cardiovascular mortality or other serious adverse reactions.
Drug manufacturing. Manufacturers emphasize successful industrial biocatalysis, which requires the use of enzyme in an effective way. This is characterized by cheap costs in high quantity of enzyme and stable enzyme in the presence of high concentration of substrates. The enzyme used in the manufacturing of statin is deoxyribose 5-phosphate aldolase or DERA. It is an enzyme typically derived from bacteria Escherichia coli or E. coli used to add the chiral side chain to the statin drug molecule. Industrial production of statin need to produce a high amount of statin as possible. However, large-scale production of statin can't afford to consume high amount of DERA enzyme because it will require an inflated cost and formation of byproducts. To overcome this issue, scientists collect DNA from various sources in the environment in a genomic library. This collection will then be screened through a fluorogenic screen test and undergo retroaldol reaction to produce an improved DERA enzyme. Another problem is DERA enzyme is halted by chloroacetaldehyde, a potent electrophile used in the formation of statins. Chloroacetaldehyde and DERA enzyme are both necessary in producing statins but nullify each other's ability. This was overcome by applying slow feeding of substrates in a certain period. Chloroacetaldehyde is added gradually at the same rate as it is consumed in the reaction. Therefore, it will not reach the concentration where it will inhibit DERA enzyme. Thirdly, barium carbonate and bromine are used in laboratory preparation of statins. However, they will cost more and cause pollution if used in large-scale production. Manufacturers replace them with aqueous sodium hypochlorite and acetic acid.
The other drug will prevent the elimination of statin, which will lead to high level of statin in the blood. Thus, muscle cell breakdown occurred and will release the protein. So, additional studies are required for better understanding regarding the role of coenzyme Q10 supplement in prevention and mitigation of statin adverse effects. Next is poor bioavailability. This is because of post metabolism, pre-systemic metabolism, and also susceptibility to effects mechanism. Yet also because of poor solubility and low permeability.
The most common approaches are bottom-up and top-down techniques by reducing the particle size through milling or mechanical micronization process. An alternative to milling is growing the particle from a solution to the desired size range under controlled conditions such as by spray drying, solvent diffusion, and supercritical flake technology. These methods help to enhance the dissolution rate by inclusion of surfactant or increasing the stability of amorphous materials by incorporation of sugars. The innovation of statin have been developed by Codexis. It creates designer enzyme to improve the manufacture of the key building block for Lipitor. The key chiral building block in the synthesis of atorvastatin is ethyl R-phosphonate 3-hydroxybutyrate, which is known as hydroxynitrile. Codexis designed a green hash-end process around the exquisite selectivity of enzymes and their ability to catalyze reaction under mild and neutral conditions to yield high-quality products. In the first step, two of the enzymes catalyze the enantioselective reduction of an ethyl 4-chloroacetoacetate by glucose to form an enantiopure chlorohydrin. In the second step, the same enzyme catalyzed the novel biocatalytic cyanation of the chlorohydrin to the cyanohydrin under neutral condition. The new enzymatic process reduces waste, uses less solvent, and requires less processing equipment. The process also increases yield and improves worker safety. Here's some extra information on effect of cholesterol and healthy lifestyle tips.